The overall goal of this procedure is to identify key improvements to the classic Western blotting technique to produce better resolution, quality, sensitivity, and accuracy of Western data. This is first accomplished by preparing samples with a buffer containing lithium dodecyl sulfate to provide the optimal condition for protein reduction and alkylation while preserving protein integrity. The second step is to separate proteins using the Bis-Tris gel system that completes electrophoresis in 35 minutes. Next, the iBlot dry blotting system is used to transfer proteins to the membrane in 7 minutes. The final step is to detect proteins using infrared fluorescent imaging that utilizes near-infrared dyes in 2 different fluorescent channels to simultaneously visualize 2 different antigens on the same membrane. Ultimately, these novel modifications of the traditional Western blotting method results in more accurate qualitative and quantitative data while greatly reducing the performance time of this technique.
The main advantages of this technique over the existing Western blotting methods is the improved efficacy, consistency, and sensitivity of Western data that allows for the accurate quantification of multiple target proteins on the same membrane, while significantly reducing the experimental time of performing this technique.
To begin, place 15 milliliter conical tubes and near confluent 10 centimeter square cell culture dishes on ice.